Thank you very much, Stephen, for the kind introduction, and how wonderful it is to be here back in Bangkok after so long. Of course, this symposium has been continued in a virtual format, but it's the first time this is hybrid, and when I had the opportunity to come back to Bangkok to see old friends, Dr. Anshali, Dr. Prapan, and many others, I definitely couldn't say no. And here we are in a very, very different time than when we last met in 2019. So today I'm going to speak to you, as you heard, about the role of antivirals in the COVID response and in future pandemics. 3 years into the pandemic, it's now clear that there are many important roles of antivirals in our pandemic response to COVID-19. We now know that antiviral treatment can prevent an individual's risk of acquiring infection. We know that antivirals can prevent an individual's risk of acquiring disease and hospitalisation. And we also know now that antivirals early in the course of disease can reduce disease severity in hospitalised patients. This is no surprise, of course, to HIV physicians who have managed the HIV pandemic entirely with only having antivirals. But we know this can be achieved with antibodies. We know that this can be achieved with oral antivirals and infused antivirals for COVID-19. But it took some time to get here. So today I'll speak to you about these current direct-acting antivirals for COVID-19 and how best to use them, why antiviral development was so delayed for COVID, talk about some new technologies that may enhance our ability to develop antivirals at speed, which is actually incredibly important in the setting of an acute pandemic, and talk specifically about RNA targeting therapeutics, and finally just speculate on what's needed for the future to change the situation should we ever be in this position again. So I think all of you are very familiar with this outstanding summary of the natural history of COVID-19 from Tixagevimab, published in early 2020. And we now have many interventions that can be used at different stages of the natural history of COVID-19. Now in Australia, we established quite early something called the Australian COVID-19 Evidence Task Force, which rapidly reviewed the enormous amounts of evidence that was coming in quickly, and yet that task force has made recommendations on when and where antiviral or anti-therapeutic should be used. So in the right-hand side of the schematic, where we're talking about severe disease, there are multiple interventions, some directed to the virus, some directed to the host, and I won't be talking about these today. The focus of my talk is what we can do early in response to acute infection, how to prevent and how to treat early disease with antiviral treatment. And we now have a number of options. Really, at the moment, the main antivirals that are currently being used, at least in Australia, are Paxlovid or Nermatrelvir, ritonavir, to some extent inhaled budesonide, and intravenous remdesivir. So I'll start with talking about these three direct-acting antivirals. And this slide summarises, I'm sure, what everyone knows in the room, the three currently available direct-acting antiviral drugs and the licensing studies for remdesivir, which is given intravenously, an RNA-dependent RNA polymerase inhibitor, Nermatrelvir or ritonavir or Paxlovid, a protease inhibitor, and molnupiravir, which has a similar mode of action as remdesivir, an RNA-dependent RNA polymerase. Now, these three licensing studies essentially looked at the same groups of people, people with mild to moderate symptoms, outpatients, and with at least one risk factor for disease progression, whether it was age, BMI, or other comorbidities. They were given early after symptom onset for Paxlovid and molnupiravir within 5 days of symptoms and remdesivir within 7 days of symptoms. And they clearly had efficacy in reducing the risk of hospitalisation or death. Quite significant efficacy for remdesivir and Nermatrelvir, about 87 to 89%, and the lower but some efficacy of molnupiravir in reducing the risk of hospitalisation or death by 30%. But what have we learnt since these original licensing studies? So the most important question for Paxlovid was, what was the real-world activity like outside a clinical trial? What was the efficacy in vaccinated people, as all those initial studies were in unvaccinated populations? What was the efficacy in younger people with or without comorbidities? There have been a number of large population-based observational studies published from Canada, from Hong Kong, from Israel, and now many other countries. I'm just going to review the data that came from Israel. And this was a retrospective case review of people over the age of 40, a huge study, about 110,000 people, to estimate the association of Paxlovid with hospitalisation and death in COVID-19, adjusting for socio-demographic factors, coexisting conditions, and previous COVID immunity status. And this is what they found. On the left was the risk of hospitalisation in people over the age of 65, almost exactly like the licensing study, a reduction in hospitalisation and death by about 90%. And on the right shows you that in a younger age group with between 40 to 65 years of age, actually minimal benefit in reducing hospitalisation or death. And importantly, they showed that this effect in individuals over the age of 65 was present in both vaccinated and unvaccinated people. So Paxlovid remains an incredibly useful intervention for people aged over the age of 65 or with comorbidities in both vaccinated and unvaccinated people. And the real challenge here, at least in Australia, is implementing its use, making sure it gets to the people who need it. What about Paxlovid rebound? There's been a lot of discussion about this in the lay press, largely because there were many high-profile cases of Paxlovid rebound, President Biden, Tony Fauci, and for those that remember David Ho from the early days of HIV, David is actually the second person listed on this case series in the New England Journal, 69-year-old man that got COVID while visiting France. So this figure shows you the pattern of Paxlovid rebound. The orange squares indicate COVID positivity, and you can see in these three cases, COVID positivity in the first 5 days. Paxlovid was administered, people became COVID negative by PCR or antigen test, and then there's this rebound at about day 10. And first point is that rebound is very clearly drug-sensitive virus. This is not breakthrough drug-resistant virus. This was very clearly shown in this case series led by Dr. David Ho. However, it's not that common. There's been subsequent large retrospective case series looking at its true incidence. And this is one example of a study being done that was done in the US, over 13,000 adults treated with either Paxlovid or molnupiravir within 5 days of their COVID infection. And they actually documented the incidence of rebound post-Paxlovid at 7 days, about 3%, and at 30 days was 5%. So this is higher than what was initially reported in the licensing study. Paxlovid rebound did happen in the placebo arm, about 1% of people. It seems to be higher in people with comorbidities and also with a history of vaccination. Does this mean we shouldn't use Paxlovid? Absolutely not. This is not common. It is not drug resistance, and Paxlovid has a big clinical benefit. What about Paxlovid resistance? Well, this has not yet been described in vivo, but has been generated in vitro. This slightly complicated figure shows you all the different mutations and their frequency if you try and grow SARS-CoV-2 in vitro in the presence of Paxlovid. And the main message here is that there actually are multiple pathways to resistance, at least in vitro. The most significant mutation is a mutation in the protease called E166V, which confers almost 100-fold resistance to Paxlovid as well as other protease inhibitors. Again, as we learnt in HIV, drug-resistant mutations can actually have a fitness cost, and E166V does have a fitness cost, which can be overcome with compensatory mutations, a principle that we are all very familiar with in HIV. There are, however, many other pathways in order to achieve drug resistance. And reassuringly, this has not yet been described in vivo, but I think it's only a matter of time that we will learn about drug resistance, particularly in using Paxlovid in immunosuppressed individuals. Some very exciting recent data that I suspect Gail may talk about more is the impact of Paxlovid when given early in disease on post-acute sequelae of COVID or PASC or long COVID. And this, again, was a retrospective study, this time done in the US at Veterans Affairs. They looked at people that had SARS-CoV-2 between March and June of 2022, that were not hospitalised on the day of the positive test, had at least one risk factor for progression to severe COVID-19 and survived the first 30 days after a SARS-CoV-2 diagnosis. And they compared the long-term outcome, or at 90 days, of those treated with Paxlovid within 5 days of the positive test, about 9,000 participants, and those who received no COVID treatments. And I think this finding is very interesting. It shows here the survival, the likelihood of post-acute sequelae of COVID in those that received Nermatrelvir or those that received no antivirals. And you can see that there's a significant reduction in the incidence of PASC by about 74% in those that received antivirals at onset of disease. Now, how does this work? We do not know that. Maybe it's because if you reduce that antigen burden of SARS-CoV-2 early in disease, you've got less likelihood of persistence of antigen. We don't know the answer. This hasn't been yet replicated in other studies, but is somewhat encouraging about antivirals having an impact on long COVID. Molnupiravir and its use has recently been quite controversial and debated actively, at least in Australia, largely as a result of this recent study published in The Lancet coming from the UK called Panoramic. And this was a multi-centre, open-label, adaptive, platform randomised controlled trial, much larger than the licensing studies, 25,000 participants, slightly different inclusion criteria, younger people aged greater than 50 or over the age of 18 with comorbidities, again unwell for a short period of time, less than 5 days with confirmed COVID, and randomised to either usual care or usual care plus molnupiravir. So a slightly different design to the licensing study. But in this study, there was no difference in hospitalisation or death across all of the study, including high-risk groups that were participating in the original licensing studies, and perhaps some benefit in immunosuppressed individuals, but this didn't reach statistical significance. The overall incidence of hospitalisation and death here was low, about 0.8%, because this study was being done in younger people and in the setting of widespread vaccination. There was, however, a reduced time to recovery in people that received molnupiravir, and a reduced viral load in nasal swabs and a more rapid clearance. And as a result of this study, the recommendation currently in Australia at least is that molnupiravir is not recommended to be used in people with mild to moderate COVID-19. Now, what about antibodies as antiviral agents? You may remember that antibodies were developed very quickly in COVID. We had the first licensed antibody from Lilly for treatment of COVID-19 at about 6 months or middle of 2020. And just to remind you about how these antibodies are generated, because it's a very important and potentially useful platform for the future because they can be made so quickly. And I also want to point out the role that HIV research played in our ability to rapidly develop COVID antibodies. This technology was developed in making HIV broadly neutralising antibodies, which we don't yet have for therapeutics, but obviously have been testing for in the setting of vaccination. So essentially, someone with COVID-19 makes a good antibody response. You can then fish out those antibody-generating cells by using an antibody to the S protein, and you can pull out memory B cells that express the S protein. You then can use single-cell RNA sequencing to get the antibody sequence, and then you can generate those antibodies in vitro, and they become antiviral drugs. You can test their potency exactly like you would test a new antiviral drug. And as I said earlier, these were developed very rapidly for COVID. Middle of 2020, Lilly had an antibody, Regeneron had an antibody. This shows you their potency against the original strains of COVID-19 in blue and in pink. And you can see these nice dose response curves with a low EC50, a concentration that will inhibit 50% of growth of the virus. But already back in the days of beta, which was in mid-2020, we could already see these antibodies had lost potency. The original formulations were intravenous. They now, of course, can be given subcutaneous and intramuscularly, which is a great advance. And the short answer now is that few or no antibodies remain active against new Omicron variants. So their utility for both prevention and treatment is limited. I will, though, point out that Evusheld, a combination of two antibodies, has clearly been shown that it can prevent COVID-19. And this is one example, a phase 3 placebo randomized controlled trial of Evusheld given intramuscularly as 2 separate doses. In this study, very small proportion of participants were immunocompromised, and the study was ended early and unblinded as vaccines became available. So it was done early pre-vaccination, and you can see here very clearly that the administration of this Evusheld antibody prevented the onset of SARS-CoV-2 PCR-positive symptomatic illness with a very high relative risk reduction of 76%. So this antibody, although not active or has reduced activity against the newer Omicron strains, is still recommended, at least in Australia, for an additional prevention strategy for immunocompromised people. So what do we know about antibodies and current Omicron variants? Well, in brief, the story, the news is not good. Here, just showing you in different colours the pattern of emerging Omicron variants, detailed on the right, going from BA1 to XBB to BQ variants. This whole swarm of new Omicron variants that we're learning a lot more about. You can see from these pictures the pattern is different across the world, the mix of variants is different across the world, and some of the Omicron variants have a growth advantage, which is why they become the dominant variant. And in this study, and this is only one of several studies, the investigators looked at these Omicron variants, either straight from the patient or by introducing key mutations, and they tested those variants against multiple different antibodies. So each antibody is a different column, and each variant is a different row. And the rows down the bottom, if you can't read it, are BA2, XBB, and XBB.1. So some of the more recent Omicron variants. And just if the box is white, it means that the antibody has no effect against the variant, and if it's dark red, it has some potency. And you can essentially see that across the bottom of the graph, there's more and more white, meaning that most of the antibodies no longer work. And the 3 that have been in widespread clinical use at different times and available in different countries, of course, bebtelovimab, sotrovimab, Evusheld, no longer work against the newer variants. So antibodies, as good as they were and being developed very early, have really struggled to keep up with variation. They're still very expensive, they're difficult to administer, and we've got a long way to go to make them better. So why was antiviral development so difficult for COVID? Well, first of all, the pathogenesis of COVID is complex. It took us a while to work out these two phases of illness, viral replication early, immunological response late, and therefore antivirals needed to be given early. In contrast, what we did with HIV is we gave antivirals to the sickest people, they worked, and then slowly gave them earlier and earlier. And of course, this took time. The current available technology for antiviral development, which means high-throughput screening of drugs, or what we call rational drug design, once you know the structure of the target, that all takes time. And in COVID, we took some wrong turns because some of this screening was done in animal cell lines where drugs like, I hate to say it, but ivermectin and lopinavir and others all worked, but when you put that in human cells, they no longer worked. We have no really good models of highly active antivirals for respiratory pathogens, very different to blood-borne viruses. And actually, very little money has been spent on developing new platforms for antiviral drug development. We did some modelling recently to look at the investment in vaccine development for COVID-19. This is not clinical trials. This was in October 2021, but $137 billion had been spent on COVID-19 vaccine development and about $7 billion on antiviral. So there's been a very big gap in investment. And this just shows you the timelines. This is Paxlovid. It's a slightly complicated slide, but the top row is vaccine development of mRNA Pfizer vaccine. The bottom is the development of Paxlovid by Pfizer. And you can see it took about 12 months after the registration of a vaccine for us to have the first effective direct-acting antivirals. And again, we did some modelling about what if we had a really effective direct-acting antiviral 6 months into the pandemic. We did some modelling and estimated this would have averted about 4 million deaths. So can we do better, and are there better technologies for the future? Well, one potential approach is that instead of targeting the enzymes in a viral replication cycle, like we do in HIV, we do in COVID, targeting protease, reverse transcription, RNA-dependent RNA polymerase. What about if we could use direct-acting strategies against the viral RNA itself? And of course, this is now possible with gene editing and also with gene silencing. Some of you will know Emmanuelle Charpentier and Jennifer Doudna, who won the Nobel Prize in Chemistry in 2020 for defining CRISPR-Cas9, gene editing technology that can target DNA. And this just shows you how it works. You have the Cas19 enzyme, you have a guide that recognises the target of interest. Here the guide is shown to you in orange, and therefore it activates Cas9 and you get destruction of the target DNA. Well, there are similar Cas proteins that can also target RNA. One of those is CRISPR-Cas13, and we've been doing some work using CRISPR-Cas13 as a direct-acting antiviral agent. I'm just going to show you very quickly. If you express Cas13, which we did here, you express the relevant guides that recognise parts of the SARS-CoV-2 virus. Here we targeted spike protein, and you have the target here, which expresses a green protein. So if you destroy the target, you lose the green signal. And what we showed was that you could use guide RNAs targeting COVID that destroyed the target RNA, and they were highly potent in multiple cell lines and also with live virus replication. So this is a proof of concept that you could use a very different approach to targeting a new virus. All you need to know is the target sequence. Now, the challenge with these sorts of technologies is delivery. But there's been actually some really exciting advances in delivery of CRISPR and other similar molecules, actually using mRNA and lipid nanoparticles, exactly the same technology that developed effective COVID vaccines. And the first clinical trial using CRISPR-Cas9 delivered as mRNA and lipid nanoparticle was published for a rare genetic illness, amyloidosis, earlier this year. So we're very interested to investigate whether you could deliver these sorts of interventions that target the RNA itself directly by delivering these lipid nanoparticles instead of into the muscle or intravenously, whether you could deliver these directly to the lung through an inhaled route. And we've been looking at this, in fact, in animal models, and this just shows you that if you deliver a labelled lipid nanoparticle into the nose and then look at the distribution in the animal model, here a mouse, you can see there is uptake in the nose, there's uptake in the lung, and absolutely nothing gets systemically absorbed in the liver, and that these lipid nanoparticles are functional. Now, this is just one approach for targeting COVID RNA or other pathogens. There are other approaches that other groups are developing, siRNA, and other ways to inhibit RNA replication. Another approach is to develop much better antibodies that are active against a whole family of viruses. And COVID, or SARS-CoV-2, is part of this family of sarbecoviruses that were found in bats, found in pangolins, and ones that are evolving rapidly as we speak. And very early after vaccination became available, a really interesting report came out from Singapore, and this was in the impact of COVID vaccination on people that had previously survived SARS-1. So their immune system had been primed with SARS-1, and then they received a COVID vaccine. And what the investigators found was that these individuals made brilliant antibodies that recognised all different viruses from the sarbecovirus family. Again, very analogous to what we have seen in HIV, that some people make really good broadly neutralising antibodies. And it's the same in COVID. These have also been fished out for people that are just vaccinated or infected or both from COVID. Here's another example of some antibodies. One of these, on the far left, 2-36, active against all different types of sarbecovirus strains. And so if we had these on the shelf ready to go, this would make a real difference in our armamentarium against future pandemics. So what's needed for the future? Well, absolutely we need more investment in new technologies. Our current approach to developing direct-acting antivirals takes too long. And there are many initiatives around the world now trying to advance this area. The NIH recently invested over $500 million in 9 centres working on direct-acting antivirals for their target list of viruses. Here you can see coronavirus at the top, influenza not on this list. The Bill and Melinda Gates Foundation, together with Open Philanthropy and Novo Nordisk, have launched a pandemic antiviral discovery program, initially for paramyxoviruses, more recently with influenza. And recently we received a gift from Jeff Cumming of 250 million Australian dollars over 20 years to set up a centre called the Cumming Global Centre for Pandemic Therapeutics, which will focus on these technologies that could be rapidly adapted, and so that in the future we will have not only vaccines quickly, but antiviral therapeutics as well. And there's of course a lot to be done about how you deliver any sort of technological advance. We will need better vaccines, better therapeutics and diagnostics, but they need to be available to everyone. CEPI, which of course did a brilliant job in developing vaccine technologies, have committed to their 100-day program, which means that they aim to have a vaccine to the next disease X or new pathogen available and delivered in 100 days. But even if we had that technology, we've got to solve this problem of equity. Just with vaccines, only the amazing statistic, 25% of people in low-income countries have had 2 COVID vaccines for a whole range of reasons, not just because of manufacturing availability, vaccine hesitancy, and many other things that all need to be solved. And it was wonderful to see the G7 recently announce their 100-day mission, which actually applies to vaccines, therapeutics and diagnostics, that we need all 3 within 100 days of a new pandemic. So in summary, antivirals clearly play a very critical and important role in a pandemic response. We have all lived that for the last 40 years with HIV. We developed antivirals slowly with COVID, and we need to do better. At the moment, the preferred treatment options are Paxlovid and remdesivir. Molnupiravir now no longer recommended. And the real major challenge here is cost, effective implementation, and ensuring that people know about how these drugs work. There's some exciting new technologies that could potentially move much more quickly, that directly target RNA and could be delivered with mRNA, lipid nanoparticles, which is going to open a whole new area of therapeutics. And so we really do need much more investment in antiviral and antibody development that are active against whole virus families and these adaptable platform technologies that could be rapidly directed to a new pathogen. I just want to acknowledge all the different people I've worked with over the last 3 years in our approach on CRISPR-Cas13, and also acknowledge our funders. And I can't stand at a Bangkok HIV Symposium and not take the opportunity to remind you that IAS 2023 will be held in Brisbane, Australia. We really want high representation from the Asia-Pacific community. Abstract submissions close on January the 31st. I promise you it will be a wonderful meeting. It is winter in Australia, but it's warm in Brisbane at this time, and hope to see you in Australia in July. Thank you very much.